Hi, today's short lecture is about viral replication. I just want to slide over here to the side for a moment and say a few different things about replication in general. Right now we're talking about how humans replicate. So I can draw my little human here, and it takes two humans, right, to make an offspring. And so we talk about how those two each produce a haploid cell. So you could take basically a sperm cell and you take an egg cell, and that equals 23 pairs of chromosomes, right? So to get to the sperm cell part and the egg cell part, we had to do some meiosis up there. So hopefully you've read through meiosis by now in both parties up here. And then when they came together into a single cell like this that we call the zygote for the shortest way to think about it, then that single cell, the zygote cell, divides, it becomes 2, and then it divides, it becomes 4, and then it divides, it becomes 8, and on to be like 16. This process, 16, right, then 32, 64, blah, 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 up to trillions of cells that makes up the human. This part, again, is the mitosis part, where each of these cells is replicating its DNA. If you look across the sort of kingdoms and domains of life, you might see bacteria. All right, now bacteria do this differently. They don't have this meiotic process. What they have is the single cell like this that goes through mitosis, essentially. And from mitosis, it makes 2 cells. And then each of those cells makes another cell, so they get 4 cells. And each of those makes another cell. 3, 4, 5, 6, 7, 8. So if you think about it this way, this individual makes those 2, this individual makes those 2. We often call these daughter cells, right? And so they basically double, the population doubles with every generation, if you want to call them bacterial generations, right? But this process is relatively slow, and some of the fastest dividing bacteria really, when you're looking at maybe an hour, if they really are fast, some of the fast ones take an hour to go from one stage to the next. 60 minutes right in here, and then another 60 minutes. And so it might take several days for the population of bacteria to grow very large from a single cell. What I want to talk about today is another group called viruses. Now, even though we don't technically call them living things, they certainly are important. And there are many different kinds of viruses. But what they share in common is that they use the host machinery. And by machinery, what I mean is enzymes, typically, to do the replication of the virus itself. And so, there are a lot of different ways that viruses can exist. So most of them are, I'll give you a couple of examples. You can have a single-stranded DNA virus, ssDNA. You can have a double-stranded DNA virus, right, which looks like a piece of DNA. You can also have a single-stranded RNA virus, right, which is just called an RNA virus. There are some other categories as well. I mean, these are the big ones. RNA typically is not double-stranded, so you have single-stranded or double-stranded DNA, and then RNA viruses, and they can be positive or negative sense, and they can be stored in different formats. And the way that viruses are classified is how do they get to the mRNA stage, right? So basically, from wherever you are, you have to get to make messenger RNA from it, and there are some different ways in here too. That is how viruses are classified. I'm going to give you an example today of a really important virus, which is the HIV virus. HIV is responsible for the largest number of deaths, the greatest epidemic on the planet, far outstripping, of course, COVID in terms of the total numbers of deaths. We're over, I think it's at 38 million people on Earth have been recorded to have died of HIV since they started discovering what HIV was in about 1985 or so, 1983. So let's take a look at the life cycle of HIV. as we go into this example, I guess I've given you this part up here, which is that bacteria reproduce by fission and viruses reproduce much more quickly using host cell enzymes. There's also a summary here of what I'm going to talk about, if you want to look through that, and it just tells you the major steps that are occurring here. Some of you may have had an opportunity to study this previous to now, and there's a lot of other steps in here that are smaller steps we could emphasize, but I think these really take care of the major components. As an aside, there are some really excellent animations of how this works. The IWASA lab, and I'll write that up, IWASA at University of Utah, does a tremendous job of visualization of biological processes, and you can check those. There's also other ones online. I'm going to begin this lecture, though, with this being my example of HIV, the virion HIV. Now it's enveloped, they say, so it has this envelope on the outside, and on the outside of the envelope it has these little spikes. And yeah, it's true, these little spikes are very similar, I guess, to those on COVID. I never used to pay much attention to the spikes until the COVID thing came along, but so sure, you've got some spikes on the outside. They're not the same as the COVID spikes, but they are still spikes. So on the inside of this, we're going to have some material that looks like this, and this is just a piece of RNA, and that means single-stranded, right? You could still get the complement of this pretty easily. So you have a single strand of RNA, so it's going to be relatively short. Small little thing, and then you're also going to have some other things in here, packaged in here, which are called enzymes. And these enzymes, sorry, I'll just call them proteins. Proteins make up enzymes, right? And in particular, the protein in here of interest is going to be one called reverse transcriptase, which comes in with the virus. So here in my drawing, I have this little virion here coming in, and it's approaching a cell. And the way it gets into the cell is a place called the Cd4 receptor. So these are little receptors that help cells communicate with each other. And it turns out it's pretty hard to get through the phospholipid membrane of a cell without having a pathway that opens up. So the spike binds to the Cd4 receptor, and that allows the cell to gain entry. All right, once it comes in, we have a single strand of RNA virus in there, and we also have with it reverse transcriptase, which does the job of making a complementary strand, right? So this is really pretty amazing. When I first heard about this, I was kind of stunned because, of course, I learned what I'm going to call the dogma down here. Dogma, right? The dogma says we go from DNA to messenger RNA to protein, right? And we transcribe here. This is the transcription area, and this is the translation area getting to proteins. Well, it turns out that if you in viruses, they bring along with them their own sort of tools, in this case, which is reverse transcriptase. And reverse transcriptase can take RNA and make DNA out of it. So that's crazy, right? So I'll just write that in. Reverse transcriptase makes DNA, and from the RNA virus, right? So you can imagine how that happens. If I do it in blue here, first you make a complementary strand, and then that complementary strand out here can utilize the host's own enzymes, and where that happens is not that important in here, but it can use the host enzymes to go ahead and fill in the other side. So now it becomes a strand of DNA. So the viral DNA then travels and goes into the nucleus. Woohoo! It goes right in through the nucleus, just like a small piece of RNA would come out of the nucleus. This one goes in, right? And what's crazy about HIV is it actually goes into the genome. So it inserts itself. Viral DNA inserts, like it basically snips open your DNA and slides into host, which is, human DNA. So now it's like you've got a new gene in here, a region of coding region, and that is the HIV gene. So in some ways, we think about HIV when people talk about, vaccines and things changing their DNA, well, that's not very accurate. But in this case, if you do get some kinds of viruses like HIV, the virus itself, yeah, it goes into the genome. And now what happens is then host, I'm going to go back to writing in black here, host DNA polymerase, right, which we've talked about before, basically transcribes messenger RNA, which really is HIV virus, and some messenger RNA that codes for reverse transcriptase. So the idea here is that you didn't have this gene for reverse transcriptase, but now you do in this cell, right? So now what happens is your body does this really fast in the same way that you would express a whole bunch of kinds of proteins for digesting food or enzymes for digesting food or to grow hair. Basically, off of here start becoming whole large quantities of like these viral particles that begin leaving, and they're all viral RNA, right? And then what you also have is some of these products get translated, right, into through the whole use of the ribosomes. We get reverse transcriptase gets made out there as well, and I'll put that out there as these little balls. So that is, you get reverse transcriptase, right, and this stuff out here is of course viral. And then these things eventually come out to the cell outside, and they get repackaged together into their own virions, right? Like make these little structures around them. And then they rupture out through the cell wall, taking a piece of it with them in this process that's called budding. And sort of the budding process ends up sort of looking like the cell wall opens like this, and they depart on the inside with some of their own stuff, and they drift off, looking like a regular piece of cellular material on the outside, because they've taken some of the cell membrane with them, and they leave. Now this can happen literally by the millions. And so when you look at the numbers here, viruses can reproduce really, really quickly. So a couple of things have happened. These move on to another cell. Why is this important for our class? Do you need to know the 6 steps? Yeah, you do. And it's important because when we talk about inheritance, so to speak, like the next generation of viruses, from a single cell, they're going to create millions of virions, really different than the bacteria process. But you could also see that from this single virion that came at the very beginning, and I guess I can highlight that in yellow, its DNA, its RNA is represented, gone into here, and it's coming back out, it's out here. And we're going to recognize that any mistakes that get made between the original copy that came in and the copy on the way out, those mistakes will get preserved in those descendants. So this little descendant guy out here will go on to infect a new cell and have a bunch of descendants. And so whatever properties are in the genes here, it keeps those. The mistakes are passed on, right? They can be passed on through this guy here. So I guess that's important. But this is, it's just a different way of inheritance working. And you know, it would be foolish to ignore viruses, even though we don't call them living things per se. But here they are. Thanks for tuning in. We'll talk again later about how treatments can work for this. I think it's also important to recognize the role of the new vaccines and how it's similar to this or not. Oh, I should point out, yeah, that some people, you know, so 5 or 6 years ago, people were looking at ways to change the DNA of mosquitoes, for example, to stop them from reproducing and kill them, basically. And they're also looking for ways to deliver gene products into cells. And so if you wanted to deliver a gene, like you wanted to modify somebody's genome, they would describe that process as being similar to using HIV. So if you want to modify the genome, right, could you use this? And yeah, you could use an HIV-like tool. Now I say HIV-like because what they like to do is take the properties of a thing that can insert itself into the genome, right? But so far it just really hasn't worked because it's really hard to do. Because you can't use HIV, but you need something similar to HIV. And the thing that was discovered that actually took the place of that is the CRISPR-Cas9 system. And we'll talk about that system, which does allow genome modification. But, you know, I had people, my cousin called me once and was like, hey, I hear they're putting HIV into mosquitoes. And I'm like, you know, it's like HIV. Those people just don't understand that most people don't know what HIV is, and who wants that in their mosquitoes? But that was never the plan. It was a system like this to use a virus to deliver a gene into the genome. But like I said, never really got it to work.